Hi, my name is June Chelsea Lee from STEM 1208. I have a mission to make a diorama of a bacteria cell from recycled materials and label its parts as well. However, there's a reason behind my mission as to why I am tasked to do this. The fundamental cause of this is to cite the importance of the cell and its unique structures. So come and join me as I participate in my journey. Here are the basic materials I needed to complete my diorama. Let us start making the body of the diorama by tracing the oval container on a styrofoam. I made two oval-shaped styrofoam.
Could you come closer to me? I just can't resist you. I'm giving up. I'm giving up. I am stuck in a dreaming. I can't help what I'm feeling. I don't know if I'm breathing when you are with me. My heart is beating. I am stuck in a dreaming. I can't help what I'm feeling. I don't know if I'm breathing when you are with me. My heart is
Let's glue them all together. After that, let's glue the other half of the diorama.
I'm giving up. I am stuck in a dreaming. I can't help what I'm feeling. I don't know if I'm breathing when you are with me. My heart is beating. I am stuck in a dreaming.
By using a pencil and a ruler, let's form the bacteria cell diorama's inner part to emphasize the cell wall.
Baby, you are on my mind.
From a scrap box, we will use this as the bacteria cell diorama's
Can't control
cytoplasm.
my body. I am stuck in a dreaming. I can't help what I'm feeling. I don't know if I'm breathing when you are with me. My heart is beating. I am stuck in a dreaming. I can't help what I'm feeling. I don't know if I'm breathing when you are with me. My heart is beating. I am stuck in a dreaming.
Now, let's wrap the diorama with tissue and paper by gluing it together to mold it into a perfect oval.
Have the strangest feeling to know what to believe in. 'Cause every time you touch me, I can't
And
control
this
my
is our
body.
finished product.
Baby, you are on my mind. All I think about, and I don't wanna stop. I don't wanna stop. Could you come closer to me? I just can't resist you. I'm giving up. I'm giving up.
Color time! I use the three primary colors to emphasize the different structures of the diorama.
Don't know if I'm breathing when you are with me. My heart is beating. I am stuck in a dreaming. I can't help what I'm feeling. I don't know if I'm breathing when you
To label the parts inside the cell, I use buttons for the ribosomes, cut-up straws for the plasmids, and a string of yarn for the nucleoids. Wires wrapped in paper for the pili, and styrofoam wrapped in tissue for the bacteria's flagellum. Wow, what a success! That was a long journey. Look how neatly arranged and attractive our bacterial cell diorama is. Well then, let's proceed into citing its unique and important functions. We got here the capsule. It plays a number of roles, but most important are to keep the bacterium from drying out and to protect it from phagocytosis by larger microorganisms. Next we have is the cell wall. The cell wall gives the cell its shape and surrounds the cytoplasmic membrane, protecting it from the environment. Moreover, the wall is responsible for keeping the cell from bursting when there are large differences in osmotic pressure between the cytoplasm and the environment. The plasma membrane is composed of a phospholipid bilayer and thus has all the general functions of a cell membrane, such as acting as a permeability barrier for most molecules and serving as a location for the transport of molecules into the cell. We have the plasmids. They replicate independently of the chromosome and appear to give a bacteria a selective advantage since it's not essential for survival, because plasmids have been an instrument in the transmission of special properties, such as antibiotic drug resistance, resistance to heavy materials, such as metals, and virulence factors necessary for infection of animal or plant hosts. Then we have the cytoplasm, the yellow one, here. It is a medium for chemical reaction. It provides a platform upon which other organelles can operate within the cell. Cytoplasm is a thick solution that fills each cell and is enclosed by the cell membrane. It is mainly composed of water, salts, and proteins. Then we have the bacterial flagellum, located here, this one. It is a hair-like structures that provide a means of locomotion for those bacteria that have them. They can be found at either both ends of a bacterium or all over its surface. The flagella beat in a propeller-like motion to help the bacterium move toward nutrients and away from toxic chemicals. Next, we have the nucleoid. It is a region of cytoplasm where the chromosomal DNA is located. It is not a membrane-bound nucleus, but simply an area of the cytoplasm where the strands of DNA are found. We have the pili. These are small hair-like projections emerging from the outside cell surfaces. These outgrowths assist the bacteria in attaching to other cell and surfaces. Without the pili, many disease-causing bacteria lose their ability to infect because they are unable to attach to host tissue. Next, we have the ribosomes. These are microscopic factories found in cells, including bacteria. Bacterial ribosomes are never bound to other organelles as they sometimes are in eukaryotes, but are free-standing structures distributed throughout the cytoplasm.